So students, good morning. We have with us Dr. Priya Gambhir. She is a DST Inspire Faculty Fellow at the Center of Biosystem Science and Engineering at Indian Institute of Science. And prior to joining the department in 2017, she did her postdoctoral research at Professor Kumaran's lab in the same institute. And she has also worked with Dr. Annie Soup in CNAME CNRS, that is in France, and her PhD is from IIT Bombay. She did in 2012, and her research theme is to study biological flows most often using mathematical methods and microfluidic tools. She is currently pursuing theoretical and experimental studies on red blood cell deformability and endothelial cell mechanics during flow. So with very short introduction, on behalf of Srishti and Honeybee Network, I welcome you, ma'am. Over to you.
Thanks, Mika, for a nice introduction. So to everybody, welcome, good morning. I hope you're having a great time at this really interesting workshop. And I hope that this particular talk, you'll be able to understand a little bit more of how usually things are done in a lab scale. Uh, I'll mostly be discussing how I use microfluidics to understand or to explore biological fluids. Okay, and it's going to be a very informal talk. I'm no professor or anything, so I don't really mind. I actually encourage if you stop me in between and ask questions. And I'll also keep asking questions a bit. So please, whenever you have an answer, unmute yourself and answer it. So without further ado, let's begin. So today I'll be talking on what are biological fluids. Why is it important to study them? I mean, they are there, but why do we even need to study them? And if we know that it's important to study them, then how do we go about doing it? What are the techniques present, etc. And I'll mostly discuss experimental techniques, and I'll just give a case study of how during my postdoc research developed a microfluidic tool to mimic the spleen on the chip. So what is a fluid? Anybody? A very, very simple, your high school physics. Can somebody volunteer to answer? What is a fluid?
Ma'am, fluid may be a liquid or a gas.
Yeah. There's some more details about its characteristic.
Any substance which have the flowing property.
Sure. So liquids especially are kind of fluids that flow. We can say matter which flows and also which takes shape of its container. So a liquid and a gas usually. We will mostly look at biological liquids, but usually even gases come under it. What is a biological fluid? How would you define it? So you know what is a fluid. So you have a background or a rough idea of what I mean by a biological fluid, but can you elaborate?
Fluids that may be present in our body.
Exactly. So fluids that are present in our body, and we can very broadly define biological fluid as fluids that interact with biological systems. So it need not specifically be humans. It can be any other biological system like microorganisms or even any other living system like birds or fishes and things like that. So, so some examples of biological fluids. What comes to mind when I say biological fluids?
Blood.
Like serum, ma'am?
Can you repeat? Sorry.
Serum.
Yeah, okay, blood.
Serum.
Most common biological fluid, or let's say most popular of biological fluids. What other things can you think of? Is that the only fluid within humans at least?
Blood-brain barrier that is present.
Yeah, so blood-brain barrier is actually a tissue. It's not exactly a fluid. It's a cell membrane that, okay, and let's not go into details, but
Ma'am,
fluid,
like pericardium fluid, ma'am.
sure, that is there. More common ones, like let's say when you cry, what do you see?
Aqueous
Oh,
humor.
ma'am, tears.
Yeah, tears is a kind of a biological fluid. And
Sweat.
sweat, exactly. Any other fluid? Okay, I've given one hint. When you eat something, you do have a fluid in your mouth, right?
Yes.
Saliva.
Exactly. Saliva is one. And then a very okay, there are some, yeah, mucus. Somebody says mucus on the chat. Yeah, that's a good example. Uh, then what else do we have? We have ears, urine, most common biological fluid. And then we have these slightly uncommon or unheard of biological fluids like lymph and interstitial fluid, cerebrospinal fluid, and so on. So all these are examples of biological fluids. And then I have this question: what about fish swimming in water? Is water then a biological fluid? What do you think?
Maybe when it is containing biomolecules,
So
can
okay, so you're partly correct. So when I see rivers, seas, and oceans, I won't say that it's a biological fluid. But when you're studying, let's say, a organism, a biological system within that fluid, then you will call that system, that particular area, as a biological fluid. For example, when scientists are researching how fishes swim in the water, so when they are considering that particular system, so the surrounding water to that fish will come under biological fluid. Okay. So that's why when I define a biological fluid, I say that the fluids that are interacting with biological systems, they need not always be inside the biological system. They can be around or surrounding it, influencing its livelihood. Okay. So these are biological fluids, a rough or a brief introduction. And then why do we need to study these? What do you do when you fall ill? Anybody? So when you feel that you are suffering from something, what is the first thing do you do? There's no right answer. Just give your guesses.
Consult a doctor.
Okay, you consult the doctor. So what does the doctor say? Does he prescribe you any tests? Depending
Yes, ma'am.
on symptoms.
Yes,
So
ma'am.
what are
Sometimes
these tests usually? Most commonly, what do you do for a test?
fluid-based tests generally like blood tests, urine tests.
So most commonly, the doctor, he'll look at your symptoms and they'll ask you to undergo a blood test or a urine test. I mean, they also do a physical examination, but if your cases are symptoms are severe, you are usually asked to go for a blood test. So biological fluids are very, very important for diagnosis of any disease. So if there's something wrong with your body, usually the fluids that are within your body also can exhibit certain of those signatures of the disease. For example, blood, it's used to diagnose diabetes, heart diseases, infection. So when you do a blood test, you measure your glucose, HbA1c parameters, you measure your LDL parameters, which is the lipid profile, cholesterol, etc. So that information can be used to diagnose these diseases. You also look at your platelet count and WBC count, and you come to know if there's any infection going on in the body. Most often, saliva is also used. For example, very commonly nowadays, you use a swab, a throat swab, right? Or you use a mucus swab also, but usually saliva is also quite commonly used for TB or COVID kind of infectious diseases. Then you use urine samples for diagnosing kidney, liver health, and pregnancy. Most common use of biological fluids is in diagnosis. So you can, while studying these fluids, you will come to know like how much do they change in any certain infection or disease. So knowing their properties in a normal healthy person and comparing it with a person in any kind of disease will help you in devising nice diagnostic tests. Okay. For example, right now you have blood tests for detecting diabetes. But there are lots of studies where they're trying to see if you can detect diabetes using, let's say, a sweat-based sample. So if you have a device where you just put your sweaty thumbprint, are you able to get a glycemic index from it? And then there are lots of other kind of tests that are going on where they want to do non-invasive tests, but they still have to deal with biological fluids. So you need to know the properties of all these fluids. How do they change during diabetes? So does your saliva also have certain biomarkers that are changing during a particular disease? Or is it just in the blood? So things like that will help you in deciding how to test for any diseases. So although we have lots of tests for diseases, it's always better to have more and more tests and try to make them as non-invasive as possible so that more and more people can take those tests, right? Then the other very important use of studying biological fluids is to increase our fundamental understanding itself of the biochemical, of the processes. For example, here I mentioned that fluid flow triggers biochemical processes which affect uh, the vessel, and in turn they affect the fluid flow. So what do I mean by this? So when I say fluid flow, I mean blood. So when heart pumps blood inside a blood vessel, what happens? Do you think our blood vessels are like metal tubes, or are they more like your garden pipes for water? Anybody? Uh, what do you think our blood vessels resemble? Are they stiff metal tubes that are used for plumbing? Are they similar to those, or are they like the soft pipes that are used for, let's say, gardening or watering your garden?
Ma'am,
Ma'am, seen
pipes.
all
Like,
soft pipes.
Okay, they're closer to the soft pipes.
Soft pipes.
They are not hard. They are made of muscular tissue. So therefore, they are very compliant. When I say compliant, they are quite elastic. Okay. So when heart pumps blood, it does it in a pulsatile manner. That is, it does a pump and relax, pump and relax. So when that happens, what happens to the arteries closest to the blood is that they expand to allow that sudden surge of blood. Okay. So if you see in this figure here, let me. So what you see over here is that your let's say this is the blood vessel in the usual normal scenario, and when there's a sudden surge of blood, it kind of uh, uh, expands a bit to allow that large volume of blood to pass through. Okay, so how this expansion happens is because of the blood flow. It triggers a lot of biochemical processes. For example, it uh, triggers the endothelial cells to release some sort of vasodilatant materials, which is nothing but materials or chemicals that will soften the wall further and allow it to expand a little bit more. Okay, so now when it expands like that, what happens is that the fluid flows into the tube, and because of the increased volume, it changes its velocity and shear. Okay, so what velocity and shear the wall experiences over here is different. So in normal scenario, this is the way the blood vessels work. But in case you have any sort of a disease, what would happen is that, let's say, for example, you have atherosclerosis, which is a condition of your arteries where they have fat deposited on them. And because of that, what happens is that they become stiffer, or they are not able to be as compliant as they are in a normal condition. When they do that, so what happens in this scenario then is that if you have blood flowing in, they're not able to give this kind of a gap to it to flow. And when that happens, you have higher velocities than usual. Okay. So how does this higher velocity affect your well-being? Or how much in a normal scenario would your artery expand to allow for the blood flow? All this kind of information is unknown. I mean, it's known now for blood, but it needs a lot of studies of fluid flow to understand how much. So, what kind of velocities is your arteries experiencing? What kind of flow profile are they experiencing? And if they are experiencing this threshold of shear, what biochemical processes are being triggered? So, all these interconnections with the fluid flow are to be understood. Okay, so to increase this understanding, we need to study biological fluids, because once we understand this, what will happen is that we will understand how a body is able to work in a homeostatic condition. Okay. Please do ask me any questions if any of this is unclear. Okay. So we know that it's important to study biological fluids for our well-being itself, and even to design diagnostic tests so that even if we are undergoing certain diseases, we can detect them quickly. It's important for that. So how do we study biological fluids? So there, for any system to study, there are two ways you can do it. One is using experiments, and the other is using purely mathematical models. So in the same way for biological fluids also, you can study them using experiments and using mathematical models. So in mathematical models, you have continuum-based models and particle-based models. So continuum models are where you write equations of physics, where you have equations defining fluid flow and the pressure and stresses that it undergoes. And then there are certain things called particle-based models, which I'm not going to go deep into because it's not my area of expertise at all. But there are these two kinds of models, or two kinds of approaches to mathematical modeling. And once you have the model ready, depending on its simplicity or complexity, you can either solve it simply, like you can find an analytical solution to the model, or you can do simulations wherein you assume a certain things, or you simulate the flow of that fluid in that particular surrounding. So both these approaches are very commonly used. Analytical solution was very common in the past, where the simulation techniques were not very well developed. But nowadays, due to the high computing power that we have, simulations are much more preferred because you can add more and more physics to it. You always have to simplify the model in case you want to have an analytical solution. But in simulations, you do not have such a drawback. So you can have the system as complex as possible. So,
Excuse me, ma'am.
Yeah, please.
Ma'am, what is the difference between this biomimicking and in vitro?
Yeah, I'll come to it. So mathematical models is one approach. The other approach, which has been done since the past, since forever, is using experiments to study biological fluids. So I'm not sure of everybody's background, but let me just briefly tell you what in vivo, ex vivo, and in vitro means. So in vivo means that you study the fluid within the living system itself while it is within its own surroundings. For example, people use intravital microscopy to study fluid flow or lymph flow within rat or mice. Ex vivo is when you surgically remove one of the vessel or the lymph vessel or the fluid that you're studying of choice, while it is still attached to the animal, but only that particular vessel is outside and you're able to view or manipulate it. In vitro experiments are where you bring all those biochemicals to your lab scale. For example, in a petri dish, you can grow the particular cells in a petri dish, and you can, let's say, for example, let us assume you want to study a blood vessel. So what you can do is within a petri dish, you can grow endothelial cells that line the blood vessel, and then you can do your studies of, let's say, shear or fluid flow or your particular assays to study how something is affecting the endothelial cells. So these were quite commonly used in the past. More recently, people are using something called biomimicking. It is also a kind of in vitro study, as in it is not connected to the animal. It's not within or attached to the animal of study. It is completely outside, but I list it as different because the length scales that we come across in this is quite different. Okay. I'll come to the details. So in today's talk, I'll be discussing more about how you can use microfluidics to study biological fluids. I hope that answers your question. Is that clear enough?
Yes, ma'am.
Okay. You'll further understand as I go ahead. Okay. So what is microfluidics? So any device that lets you manipulate fluids in micron-sized dimensions is microfluidics. Very simple. Okay, and when I say manipulate, you can either hold the fluid, you can flow it to different parts, you can react it with other fluids, or you can study any properties of it. So any kind of manipulation of fluid, but within micron sized dimensions. And not every dimension of that chamber has to be micron sized. Even if only one dimension of the chamber is micron sized, your device is still a microfluidic device. Okay. So, have any of you done microscopic imaging in as part of your studies ever, or have you even seen a demonstration of it?
Yes, ma'am, we have used microscope.
Okay, so can you tell me what you do to prepare your sample for microscopy?
Like staining, ma'am.
Yeah, staining, or what do you use as a substrate for your sample? Okay, let's say you want to observe a drop of blood under a microscope. What would you do?
Ma'am, we will make a thin layer of our bed cell
Sure. So how do you make that thin layer?
that so that we can see we can observe it in the microscope.
Sure. So how do you do it? How do you make that thin layer of sample? Okay. So let me, she is completely right. What do you usually do for when you want to observe something under a microscope is that you place a sample of whatever substance or cell or chemical you want to observe. You put a drop of it on a microscopic slide. Okay, this is a glass slide, which is not at all a micron sized dimension. Okay, it has nice centimeter dimension, so it's like around 7 centimeters one side and 2.5 on the other side, something of that sort. And then what you do is after you put a drop of your sample, you place a cover slip on top of it. So you place a cover slip for many reasons. You don't want the sample to evaporate during your imaging. You don't want it to spread or interact with the dust or microbes in the environment. So you want, in order to protect it during your imaging, you do that. And also when you place a cover slip on top, it ensures that your sample is spread completely and you get a nice thin layer. So when you're doing this, that is when you take a microscopic slide, you put a drop of sample on it, and then you cover it with a cover slip, you are making a microfluidic device. This is nothing but a microfluidic device. So when I say that at least one dimension has to be a micron size dimension. So in this case, your microscopic slide and cover slip are not micron size, but when you put them together, the gap that is generated, which is holding the fluid, is a micron sized gap. And therefore, we will call this as the very basic microfluidic device that is being used. Okay. And here, if you can see this image, this is a very common microfluidic device. What they've done is they've just put a dye inside it to show the channels. So these circles over here are inlets or outlets where you can put tubing and then you flow the fluid and then it passes through all these channels and you can either pull it out from one of these outlets. Okay. Why I put this image over here is to give you an idea of how small these devices can be. So this is just a coin of some currency. And you can see that the devices, the channels are actually much, much smaller than that. To just give you a perspective of how small a microfluidic channel can be. Now, what I'll do is I'll play you some videos from YouTube where there are a few microfluidic devices in work, okay? Let's see if it works. Okay, do let me know if you are able to see the screen and the device video, sorry. Are you able to see it? Anybody can answer?
No,
No,
ma'am.
ma'am.
Okay.
No, ma'am.
So what you see over here is, this is the
No,
channel.
ma'am.
Okay, let me play it again. Okay. So this here is a channel, okay? You see red, white and blue streams over here. So this is the main channel and it has two branches. In one of the branch, they've put in red fluid. In one of this, diagonal branch, they put in blue fluid. And then there's a central channel also in which there's a clear fluid, that is colorless fluid. So all these three fluids are entering this one main branch, in which what they are doing is they are changing the flow rates of the two branches, the red and blue fluids. When they change the flow rate, if they are increasing it, you'll see that the width of this red and blue in the main channel is increasing. Okay, can you see these images that I'm just moving?
Ma'am,
Hmm.
ma'am, we are not able to see the video.
Okay, is it at least visible as a screen?
No, ma'am. Only
at
the
all?
PPT is visible.
Okay, okay, then.
Hello, ma'am.
Yeah, tell me.
Ma'am, please use on the sharing option. You have options to change
Okay.
the screen, ma'am. Then we can only see the video.
Okay, then. Yeah. Is it now visible?
Yes,
Yes,
ma'am.
ma'am.
Okay. So you can see that there are red and blue streams that are growing and contracting. Okay. Here is a microfluidic channel. Let me just go to the beginning. Okay. Now we have this red. Can you see a diagonal channel over here where my arrow is pointing?
Yes, ma'am.
Yes, ma'am.
The red branch, there's a blue branch, and there's a straight channel which contains a colorless liquid. Okay. So what is happening in this particular video is that there are three streams of fluids coming in, and by changing the flow rates of either the central channel or increasing the flow rates of the side channels, you can actually change the width of these streams in the main channel over here. Okay. And what you'll appreciate over here is that these three are miscible fluids, but yet the flow is so streamlined that you don't see any mixing. Okay. And when you change the flow rates, you can see that it's reacting immediately. So immediately you see that the red stream and the blue stream are growing thicker and the white stream is getting thinner. So you can manipulate the flow rates and change the streams in the main channel. Okay. Just keep this in mind. That will be important when I discuss the advantages of microfluidics. Then the other video is here. This is another common. Okay. Are you still able to see the video?
Yes, ma'am.
Yes, ma'am.
Okay. So there's another common example of microfluidic devices where they generate droplets. Okay. So if you see over here on my right side, there's a channel which is putting in a blue liquid, and on its either sides are two channels at 90 degrees, which are giving clear colorless liquid. And the design at this construction is such that in the previous one, they all were coming in a nice streamlined direction or angle. Over here, it's at 90 degrees, and plus the construction or the junction is designed such that when you have flow coming in from the perpendicular channel, it cuts this blue fluid such that you start forming droplets. And also over here, the blue fluid is immiscible in the colorless fluid. That's why you end up getting these nice droplets. Okay. So these are just examples of microfluidic devices where you'll appreciate that there's such high control on the flows. You're able to manipulate the thickness of streams and the flow of the central channel, and you're also able to generate very monodispersed droplets. That is, the droplets are all of very same size. So if you collect all these droplets and measure their sizes in certain equipment which are able to give you those sizes like DLS, you'll see that their sizes are really, really standard. Okay, and you can manipulate the size by changing the flow rates again. So if you increase the flow rate of the colorless liquid, you'll see that the drop size becomes smaller. Okay, so there are actually given for a particular width of the channels, you can make a calibration graph where you can say that, okay, I'm going to have this velocity and I'm going to get this drop size. Okay. So let's go back to my presentation. Yeah. Okay, so am I back on my slides?
Yes, ma'am.
Okay. So those were a few examples of microfluidic devices and how people are able to achieve flows in them. So we'll come to why are people going so gaga over microfluidics, okay? Why is it so suddenly become the keyword and all that? So the main advantage, especially for the topic that I'm discussing now, is when we study biological fluids, it's very important or microfluidics have special advantages because the first and foremost is low sample volume. Usually your blood, your body fluids, you acquire them in very low samples. Because let's say you're taking a saliva swab or a throat swab, you hardly have a microliter or so of it. So when you want to do in vitro studies with these biological fluids, what happens is that your systems are quite large. So if you're working with, let's say, a tissue culture plate or even a tiny cuvette, it's already of a very large volume. So when you put in microliters or tens of microliters of fluid, it's usually lost on the walls, or it's very difficult to get, or you will have to work with larger sample sizes. So to avoid these kind of problems, people have moved towards microfluidics. So if you look at this tiny calculation over here that I put up, you will see that in a very typical microfluidic device, here only one dimension is in the micron size. So I'm saying that the length of the channel is 4 centimeter, which is quite large. And the width is 0.5 mm. So it's still not, it's still in micrometers, whereas the height I've brought it to 100 microns, okay, the height of the channel. So when I do that, that itself gives me a low volume of 2 microliters, okay. So a large enough channel that is visible lengthwise, but its thickness is 100 microns, and it can give you 2 microliters. So you don't have to work with large biological sample volumes, and you can work with slightly lower. Then you have a very high surface area to volume ratio. So this is very important again when you're studying biological fluids, because most of biological reactions occur when they are in contact with certain substrates. So it's always good to have as large surface area as possible. So here what I'm trying to convey to you is that I have two rectangular macro channels, and only one of them has a height of 100 microns. The other has a height of 1 mm. Okay. So the length and width are the same for both the channels. And just by changing changing one dimension into microns, and that to not lower microns, just by one order of magnitude. So 1 mm is coming down to 100 microns. What I've done here is I've calculated the surface area S1 and the volume V1 of the first channel, and the surface area and volume of the second channel. And you'll see that the surface area to volume ratio for the second channel, where you have the height as 100 microns, has increased tenfold. Okay, you have so much higher surface area when compared to the volume. So even if you work with very low volume of biological fluids, you can still have lot of reaction area happening because you have higher surface area. I hope that's clear. You're able to appreciate the advantage of this. Then comes laminar flow. So I'm not sure if most of you are familiar with this term, but what it comes down to is when you, as you saw in the video, the blue, white and red streams, they were not mixing, but they were flowing in a very streamlined fashion. That kind of flow is called laminar flow. As soon as you have disturbance within these flows, it's called a turbulent flow. So within microfluidic devices, most commonly you have laminar flow, which is very difficult to achieve in a macroscopic system. So when you're working with, let's say, a flow in a large tube or something, you always see it's very difficult to hold back turbulence, or even when you're working in beakers or anything of that sort. Whereas within microfluidic devices, it's very easy to achieve laminar flow, and that ensures that you have extreme control on your system. So like I said, as soon as you change the flow rate of the red and blue streams, you see that in the main channel also, accordingly, the widths are changing quite immediately. And because it's so small in sizes, you don't have gravitational effects on the fluid. So when you have a large tube and when you flow a little bit of fluid in, what you see is that the fluid settles on the bottom of the tube, okay, or it flows along the lower wall of the tube due to gravity. Whereas in microfluidic devices, because of their low size, they have very low volumes and masses of fluids in them. So gravity does not work on them. So that means even if you put very low amount, let's say your tube height is 100 microns, but you're putting in only 1 microliter of your sample. So the sample will not sit on the bottom plate or bottom level of the microchannel, but it will fill the entire cross section. So what that gives you again is extreme control. So you can push that liquid quite easily inside the tubing. This is very difficult to visualize when I explain because we are not used to seeing this kind of system. But when you start working with microfluidic devices, you'll understand what I'm saying. But just take it for now that this is one of the major advantages of it. Then you have confinement effects because, as I said, most of the biological fluids have cells within them. Okay, for example, blood has blood cells in them, lymph has lymphocytes and flowing through them. All these cells are actually of micron-sized dimensions. So when you work with microfluidics, you can actually work or design devices where you can confine them or confine particular cells so that you can do single cell studies. This is very difficult to do in your traditional in vitro experiments. You always have to work with a population of cells. But this is a very good advantage of microfluidics while working with biological fluids, especially. And then portability, of course, because your device is small, you can carry it around. So it's quite portable. And it's high throughput because now your channel is so small, so you can have multiple channels. Because let's say if the channel width is only 0.5 mm, so on a microscopic slide, which is 2 centimeters wide, you can put lots of channels and you can have parallel reactions or studies going on. So it can be made into a very high throughput system. So these are all the advantages of microfluidics. We know that what are biological fluids. We know that one of the ways of studying them is using microfluidic devices. Now the question to ask is, how do we go upon making a microfluidic device? That, okay, I know that, yeah, microfluidic devices are very interesting. I want to make one. So how do we start going about it? There is no single design or single description of a microfluidic device. It's very customized to the study that you want to do. Let's say I want to work with certain cells. Let's say I want to work with red blood cells. So I'll make a device which will be of dimensions where I can confine a single red blood cell. So I would prefer to have those dimensions. But if I'm, let's say, working with just serum, I'm removing the blood cells before working with it. So serum is like a watery substance. There is no confinement necessary. So I can have 100 micron gap because I have a lot of serum once I take out the blood cells. So that way, depending upon your study or the problem statement, you will have to design and develop your own device. So how do we go about that? I'll give you a case study where I've made a spleen on chip, and how did I go about doing it? Okay, any questions until now?
Yes, ma'am, I have a question.
Yeah, please do ask.
You talked about like we need to control the flow rate and velocity of the fluid. So how do we
Yes.
do that?
So there are these things, very good question. Thanks for pointing it out. I should have mentioned it before. So we have lots of pumps. So just like you need a pump to push blood within your blood vessels, you need a pump to push any kind of fluid. So we have nowadays lots of pumps available. The most common are called syringe pumps. So they are these setup where you can put a syringe full of fluid, you attach it on the pump, and then it slowly pushes the plunger at a given flow rate. So you can actually go down to flow rates of microliters per hour on it. And then the syringe, the other end of the syringe, using tubing, you connect it to the microfluidic device. I'll just show you quickly. Yeah. So here, let me use a pointer here. Okay, so here you see these two holes, right? So over here you attach tubing. So you actually get tubing of those sizes nowadays, where you have outer diameters of 1 micron or less. So such kind of tube you attach to this device. And then the other side of the tube is attached to a syringe. And therefore, you can pump fluid through the syringe into this device. Does that clarify your question?
Yes, ma'am. One more question. Like you also talked about on the confinement effect. So is it related to it? And you also mentioned the single cell study. So how do we do that single cell study with respect to confinement effect?
Okay. So for example, now in this device itself, you'll see that these channels are quite large. But when you come here, you have a thinner channel. So what happens is that, let's say I have a sample of I'll tell in terms of blood because mostly I've worked with that. So what I do is I use a very dilute solution of blood, and then I pass it through this channel. And when it comes to a very constrictive channel, the population of blood itself, self-organizes. So one cell at a time will pass through that channel. Because when one cell passes through through this channel, it blocks the flow. So until the cell passes through it completely, you will not have the other cell passing through. So it will become clear when I come to a few more videos later in my presentation. Okay. Be sure to ask me this question again at the end. So if it's not answered yet.
Thank you.
Okay. Any more questions? Okay. I'll go to this case study. So red blood cells, I'll just give a brief introduction. They are very deformable cells. They are not very similar to other cells in our body, as in they do not have a nucleus inside them. They are just like an elastic capsule. Okay. And they have extremely high uses within the body, and more advantages or functions of red blood cells are still being discovered. Okay. So they look something like this. They have a discoid shape, a biconcave disc shape, wherein their diameter is around 8 microns and their height is 2 microns. Okay. Now I said they do not have a nucleus inside them. It's filled with a watery substance called cytoplasm. And their membrane has this kind of a protein lipid structure. Okay. You need not go into the details of this. Just understand that this structure is very elastic. Okay. So what this does now, because they are of this discoid shape, if you compare the same surface area of a sphere, their volume, if you compare the volume of this RBC with the same volume of a sphere, the surface area of RBC is much, much larger than that of a sphere. Okay. What that means is it has excess area for a given volume of liquid that it holds. And that allows it to take a lot of shapes. Okay. What I can give an analogy is with a balloon. Let's say consider a balloon where you have removed part of the air of the balloon. Okay, it's not completely inflated, slightly deflated. So then what you observe is that it's more easy to press into it or deform it into a different shape. Do you understand what I'm getting at? If it's a completely inflated balloon, it is much more stiffer. You find it much more difficult to push into it or deform it in any way. But if you, as soon as you let out a little bit of air from it, the balloon has excess area, that is for a given volume. That is, it has a lesser volume and it has more area. What that ensures is that you can actually press into it and deform it quite a bit. The same is true for an RBC too, for a red blood cell. And also the cytoskeleton, it has very dynamic linkages. So that helps it, get more deformable. So the biggest test of, the deformability is always tested in the use of the deformability of this red blood cell is when it has to pass through these tiny capillaries, So the red blood cell being 8 micron, it has to pass through capillaries which are 6 micron sized or 4 micron sized. So it has to actually deform so much smaller than its own size to be able to pass through these capillaries. And it keeps doing this throughout the day, throughout our lifetime, its lifetime specifically. So red blood cells are replenished every 3 to 4 months. But until that 3 to 4 months, that particular red blood cell, it keeps deforming every time it passes through these capillaries. Okay. So the most most rigorous test of red blood cell deformability comes within the spleen. Okay. So what is spleen? Anybody? Do you know what a spleen, what the organ spleen is?
One of the immune system organs.
Yes. So spleen is one of your organs of the lymphatic system. It takes part in immune function. It also is a very, very important organ to filter your red blood cells. So as I said, red blood cells are replenished every 3 to 4 months. So as soon as your red blood cell becomes old, it has to be destroyed and then replenished with fresh red blood cells. So to do this, this is tested every day, every second within your spleen. Okay, the spleen has these kind of gaps. Can you see this tiny black gap over here pointed by the arrow? So this is a
Yes, ma'am.
microscope of the spleen. So this is one of the red blood cell. This is another. So these have to pass through these really small gaps within endothelial cells of the spleen. So here is a schematic, okay? So blood enters into the spleen and then it has to pass through these tiny gaps and then it can exit into the vein. So if it is not deformable enough, so as the red blood cells become older, they lose their deformability. So the older red blood cells, they are not able to pass through these slits and then they end up remaining within the spleen and then you have these phagocytes which of the immune system which destroy the red blood cell and recycle its contents. Okay, it's a very efficient technique and it's pure filtration. So our problem statement was that we wanted to design the spleen on a chip. Okay. Now, why we wanted to do that comes in the next part, but we wanted to mimic these dimensions. Now, these dimensions are the width of this gap is around less than a micron. That is, it is 500 nanometers. Okay. And its thickness, that is thickness of the endothelial layer through which the cell has to pass is 2 microns. And the length over here that you see is 5 microns typically. So on the channel, we wanted to mimic these similar dimensions. That was our problem statement. Why we wanted to do that is because we have not seen in vitro how does RBC deform so much. Like what kind of shapes does it take while passing through this extreme mechanical tests, okay? And can we make a device to mimic this kind of filtration capability of the spleen? And would it in vitro show a difference in deformability just based on the filtration capability? So we wanted to understand all these questions. Most importantly, we wanted to see that does the RBC deform as much every time? Okay. So now here comes the question, how do we make the microfluidic device? Okay. So there's something called silicon wafers. They look something like this. This is a schematic, but most of the wafers look like this. They are really flat. They are made from single crystal silicon and they have a flat on it. So what is usually done is a technique called UV lithography. Okay. In this technique, what is done is you have this wafer, you coat it with a photoresist. Now, photoresist is a material which is it's like a pre-polymer and it can be cured under UV light. Okay, so we just coat a very thin layer of this photoresist using something called a spin coater. Then we make a mask. Okay, the mask will have the design of the micro channel that we want to. The mask will have a design of the micro channel that we want to build. So this is the UV mask design of the device. This is made using any CAD software, computer-aided design software. Okay. Usually you need to have an inlet and outlet, and you need to have some side channels in case the channel gets blocked. You can flush out or send the waste and all that. And then the dimensions of the device will be decided by the system that you're studying. So in my case, like I said, we had decided upon the channel dimensions that we wanted. Now the channel dimensions that we decided were very small. So directly putting a fluid from a macroscopic system to that kind of a micro channel is very difficult. So we begin with a 300 micron width channel and then we slowly come to the splenic channels. Okay. So this is the mask design. So here I've indicated it using this schematic. Okay. Wherever there is brown over here is where the UV light cannot pass through, and wherever there is white, UV light can pass through. Now this photoresist can be of two types, positive or negative. So depending on that, you can either remove off the parts that are exposed to UV light, or you can keep the parts that are exposed to UV light. So depending upon how much your design is and all those factors are taken into account. So basically the step is that you have silicon wafer, you coat it with photoresist, you shine UV light through your mask, so that only parts of the photoresist are cured to form hard polymer. So once we do that, we put it in a developer, which is nothing but a chemical which will dissolve the unpolymerized photoresist. Okay. So when you do that, only the parts that you want will remain on the silicon wafer. So this is called a master mold. Okay. Is this clear? This part of it? This comes under a technique called UV lithography. So once you have the master mold, what you do is something called replica molding. So replica molding is done only when you want to have a device made in PDMS. So PDMS is nothing but a polymer. It is optically transparent and it is not very toxic. So that's why it does not affect your cells or your biological fluids. And therefore, most often, this is what is used. So you make the device in PDM. To make the device in PDMS, you pour the PDMS liquid before it cures. And after it cures, you pull it out and you seal it with another glass slide. Okay. So here is what our design was. This was the channel that we planned. Then these were the tiny channels that we placed within the larger channel. So once we did our lithography, we get this kind of a design. And then when we do PDMS, so this is called a positive master. And then when you pour PDMS, wherever there are bridges, they become channels. And wherever there are grooves, they become bridges. So now this part here is empty on the wafer. So that is filled with PDMS. So it becomes a bridge in the PDMS. So is that here? Okay, it'll be more clear over here. So this is just some more information about the design dimensions. So this is nothing but this is on the silicon master mold. So this is a bridge on the silicon master mold. You can see that we are able to get different widths and different heights and inclination also. And once we do that, it forms, when we pour PDMS on top of this bridge, the PDMS will have a hole like this. So this is our actual channel through which we'll pass the magical fluids. Okay. Now I'll just quickly show a few videos of the red blood cell passing through these channels. Okay. So now here you see this is a micrograph. These rhombus things are solid PDMS, and the gap inside this is the actual channel. Okay. Now here is a red blood cell. All these three are healthy red blood cells. Okay. So we'll just see how they pass through. Are you able to see the video?
No, ma'am.
Oh.
Yes, ma'am.
Yes, ma'am.
Okay, so red blood cell has passed through the channel. It is forming a dumbbell shape while it is inside that constriction. Okay, because it is a larger volume, it fills the channel and still bulges out of it. And then it very easily comes out of the other side. And it actually also reforms its initial shape quite easily. Okay. So now all the three videos are playing. Even the inclined channels don't seem to have much difference. The healthy RBC can easily pass through these very tiny constrictions too. So then we did further studies of their recovery rates and the shapes that they form, etc. But this is just to appreciate that the micro channel is built and it is being used to study certain biological fluids, in this case blood. Then we went and took a sample of rigid RBCs that we know would be rigid, because sickle cell anemia is a disorder where most of your RBCs are lost in the spleen, and therefore you become anemic because there's low hemoglobin. Okay, so in this case, we thought that because it's anemic RBC, it would not pass through. But you see that it actually passes through quite easily, even in this case. Okay. Are everybody able to see the videos?
Yes, ma'am.
Okay.
Ma'am, how you means how you put the pressure on cells, ma'am, through the syringe or what?
Yes, yes. It's through the syringe. In this particular experiment, we had a pressure-driven flow system. That is, we have inlet outlet, and it is connected to a gas cylinder. Okay, so and through a device actually. So the instrument ensures that there's a pressure drop maintained across the inlet and outlet of the microchannel, and that will push the liquid fluid to pass through.
Okay.
Okay. Then
Ma'am, in the third video, excuse me, ma'am.
yeah,
Ma'am, in the third video, is there any deformation happening like in RBCs, like in the third video?
Yes. So you have to understand that these are sickle RBCs. So usually they form a sickle shape. Okay. So in the third one, the RBC, the initial shape itself, if you see, it is slightly elongated. It's not exactly circular, such as the one in second video. So it already has more deformability to begin with. And therefore, when it exits also, you see this really elongated shape. And I'm glad that you pointed it out because we'll be discussing that in the next slide actually. So if you look at this also in the second video, you'll see that the sickle RBC, when it exits, it forms this tip. Okay, this very sharp tip, which is remaining for a slightly longer time, and eventually it relaxes. Okay, so that really sharp tip is something which was not observed before. Now, then we went ahead with another extreme anemic disorder, which is called hereditary spherocytosis, in which the children have such extreme anemia that they actually do not survive very long. So, in this case, we saw that uh, the cells cannot pass through the splenic slits at all. So, if you see in this first video, all of them come and they just get entrapped within the channels. Okay, and they block the entire channel. And here a few of them are able to pass through, but then they form this extremely weird shape where like a jellyfish. Let me try to see. If you see here this shape, it looks like a jellyfish, and you can clearly see that the cytoplasm has moved to the front and the cell membrane has detached from the cell. Okay, so it's very difficult for them to reform into the usual shape after this. So as you mentioned, there are these, okay, very quickly, the healthy samples are transiting very easily. We have very high percentage of them transiting, which is indicated by orange. And the sickle cell anemia also, as I said, we expected that they would be blocked, but they are not. Most of them are able to transit quite easily through the slits, but it's the other way around in the hereditary spherocytosis. We see that most of them get entrapped. And even if we push them faster, we still see 50% of the cells getting entrapped. Okay. And then we see this very unique shape called jellyfish where the membrane seems to have detached. And then, as you rightly pointed out, sickle cell, although all of them are passing through the slits quite easily, there should be some effect of their anemia on the cells, right? They cannot be as good as the healthy cell and still be suffering from anemia. So what we did was we counted the number of cells that are showing these elongated deformed structures. And we saw that the sickle cells actually deform quite a bit when they exit through the spleen. And they take very, very long to relax also back to their original shape. So if they are already deformed and they have to pass back through the splenic slits, then there is a chance that they might get blocked. This is something that we did not check. We did not repass them through the slits. And therefore, we cannot comment if this will actually happen. But this might be one of the case why there is anemia within sickle cell patients also. Okay. So this one I'll skip in the interest of time. But to summarize, what we have done is we've used photolithography and other technique called wet etching and replica molding to get sub-micron size channels in optically transparent biocompatible device. Okay, so those were our key requirements for the device, that it should be optically transparent because we wanted to see the deformation that occurs at this extreme high constriction. And it had to be biocompatible because we are working with the red blood cells. We do not want them to just start rejecting the device or the material. So the spleen on chip, it can actually differentiate between healthy and diseased cells. And red blood cells, when passing, they show these novel directions. And using this data, we could define some future directions for the study. So from this particular lecture, I hope you've understood that what biological fluids are and why is it important to study them. And one of the approaches to do this is using microfluidics because it has its unique set of advantages when you're working with biological fluids, and how to make a simple microfluidic device in lab. Okay. So first thing would be to define your problem statement. What would you need from your microfluidic device? And depending on that, you choose the techniques to make the device. You define the dimensions that you want in your microfluidic device. And then once you make the device, you have to check with different samples of biological fluids if what you want is being demonstrated or not. Okay, and then you can make tweaks and further changes in it. So that is it from my end for this lecture. I'm happy to take questions or re-explain any part of it. You can also. Yeah.
Yes, ma'am. Do you face any problem, ma'am? What do you do to make such narrow channels?
Okay. So good thing you asked that question. It's a very nice question. So I did not put that slide over here, but usually UV lithography does not give you submicron size channels. So UV lithography is good enough to only give you channels of, let's say, 10 micron or higher. Even 10 micron is a stretch, okay, because the UV light, it bends and things like that. So there are other techniques like electron lithography, electron beam lithography and all that, which can be used, but they are very expensive, and it's very difficult to get access to those instruments. But UV lithography nowadays is quite common. So that's why what we did was we wanted to use the instruments at hand and still get submicron channels. Therefore, I read quite a bit of literature about what can be done, what are the different microfabrication techniques. And I realized that there's something called wet etching that is done. So what wet etching does is it's just plain potassium hydroxide, KOH, that is added to your wafer, and it dissolves part of the wafer. So you can have silicon oxide on top of your wafer, and it dissolves that silicon oxide. Okay, so then what I've understood that particularly, so if you keep doing the wet etching for a longer time, it'll eat at particular planes of the silicon wafer longer. So using that technique, actually, therefore I designed the initial master or the mask. The CAD actually had this dimensions for the submicron channels or the micro channels. So we wanted them to be 0.8 micron and 2 micron. And I began with 16 and 11 micron. So once we made this master, I put it in KOH, and KOH dissolved it such that I obtained these in the end. So I had to do a lot of experiments to fix the initial dimension so that I knew the rate at which KOH is dissolving silicon oxide. So therefore, if I put this device for 10 minutes in KOH, in the end, I'm going to get the right channels. So that was one part of the device fabrication, which was little non-trivial, which had to be worked with. I hope that is what you were looking for.
Yes, ma'am. Ma'am, one more question, ma'am. Ma'am,
Yeah.
how can we study, ma'am, cerebrospinal fluid non-invasively, ma'am?
So non-invasively, as in generally now blood also. When I say non-invasive, they are already giving blood for some other tests, and we are using that. Okay, I understood what your question is. Usually, you can use optical methods. For example, nowadays about cerebrospinal fluid, I'm not at all the expert, so I cannot comment. But for blood, I can tell you. So you can use electrical techniques or optical techniques, wherein now if you look at your phone sensors, you already measure your heartbeat and things like that
Yes,
using your just the optical technique, right? It actually looks at the pulse, the pulsing of the flow within your finger, and it gives you your heart rate. So that is one way of studying blood non-invasively. But the information that you can get is limited through that. So depending upon the test that you want to do, you'll have to see if you can do it non-invasively as much as possible. There are, of course, a lot of techniques that are used by brain researchers to do a lot of tests. For example, they also usually use electrical impulses also. But I'm not sure if they study the electrical impulses from the brain, but I'm not sure if they have studied cerebrospinal fluid specifically using any non-invasive techniques.
Okay.
But yeah, that is much open for research, that means.
Excuse me,
Thank
ma'am.
you.
Yeah.
Ma'am, in hereditary spherocytosis case, you make us watch the video of like there were the cells which are not able to pass through the channel. So
Yes.
I observed one thing like there was a cell which was going through a channel in which the cell is already stuck over there, but suddenly it changed its direction. So is it the case
Yes.
of pressure gradient created by the already
Exactly.
stuck cell over there?
Yes, exactly. So what is happening is that you cannot see it, but there's a lot of the fluid streamlines are such that now this is stuck over here, but the fluid comes over here, but then it is directed because there's pressure drop only in this direction. So the cell also follows that streamline. You're exactly right. So there's pressure gradient on either side of this, but just because there's a cell stuck here, the fluid itself is redirected in this direction. And therefore, the cell that comes over there, it follows that fluid streamline. That is a very nice observation. Good. Any more questions? Please feel free to ask even the silliest of questions, okay? This is your chance. Oh, sorry. Am I not sharing my screen? Yeah, any more questions?
So students, apart from this presentation, if you want to ask anything, any biotechnology question, any query of your project.
Yes, please. I don't know if I can answer it to your this thing, but I will try to. Please feel free to ask. Don't be shy.
No more.
Hello, ma'am.
Yeah.
Ma'am, when you showed in the second video that in the microfluidics that small molecules start forming, ma'am, blue molecules you showed.
Yeah, they're droplets.
Yes, ma'am, that droplets. So, ma'am, this type of microfluidics is also used in the protein crystallization process. So, ma'am, how do you calculate this concentration, ma'am? Ma'am, in the crystallization, multiple means oil, protein, buffer, salt, multiple things mix and make a droplet. So, a droplet or a molecule. So, how do you measure its concentration?
Acha. So that you'll have to customize it to your application. So what I showed over here is to make a basic droplet. So you can use the velocities of either of the channel to make the size of the central droplet. But once you form those droplets, you can probably test them for the concentration that you need, and then probably tweak the velocities of the other two channels to get a larger or smaller droplet. That should be one of the ways to go ahead with it. I have only worked with bare droplets, so I'm not sure how do you go about measuring concentrations. But that is one of the things. There's a lot of literature where you can look at these issues. especially droplet microfluidics, it's quite well researched right now. So probably that should
Okay,
help
ma'am.
you. Mm.
Thank you.
Anything else? Please feel free to email me in case you have any queries that you, it's just gpriya@iic.ac.in. Okay.
Okay, so
Okay, Mega. Yeah.
thank you.
Stop sharing.
Yeah. If you have any query, you can just I think you have noted down the email ID, or I'll provide you email ID in later part if you have any query. But thank you so much, Dr. Priya, for your
Thank
time.
you. It was my pleasure.
It was really a very nice presentation and very interactive session.